All right, everybody, let's settle down here. And I'm about to hand over the reins to Alexander Haig. I mean, our...
You're going to be here all week?
What?
You're going to do some shtick?
I'm not going to do any shtick. You're safe. I am very pleased to introduce the person who's going to relieve me of all responsibilities for the rest of this session. Our leader here is a graduate of HST, MD, PhD, worked with our stem cell biologists at Harvard, and then went on to be involved with several companies in the genomic space and tech space. And most recently, he has become senior partner at Takeda Ventures, and also is father of 3 lovely girls. And any mistakes he makes tonight are purely the result of his ketotic diet. Thank you very much.
That's with that diet, man. All right. All right, looking forward to this. So just before I start, this is my favorite conference of the year. I was trying to think about why that is, and I think it's the most Hamish conference I attend, which may say a lot about some of the others. But I think it's really the combination of incredibly high-quality science, but really built around an incredibly deep humanistic essence and a core, and fundamentally around the patients. And it obviously reflects the characteristics, vision, and warmth of the leadership, and of Zach in particular. So we could just sort of start by giving some recognition to Zach and the organization of this conference. All right, so this panel, we're going to get to it because it's unbelievable talent, is about hyper-personalization or hyper-personalized therapeutics. And I just kind of, one slide. Just for sort of thought, we kind of start on the one hand, it's the sort of the vision, future work into, I don't even know what that is, but that in the future, everything is going to be these hyper-personalized treatments. You show up and, some 3D printer in the back is going to manufacture exactly the right drug for you at the right time. In contrast, I thought it was worthwhile to compare this perspective from Harvard geneticist Richard Lewontin, which he wrote in the New York Review of Books in 1992, when he was sort of in a very famous essay called I Dream of Genome, which was essentially about and in his words, the very first thing he starts off with is the definition of a fetish. And he was basically making the point that scientists were fetishizing the genome, which now has arguably been extended to fetishization of data tech and all of this stuff. And he essentially is saying that there's a tendency of people to overpromise what the science can deliver. So in that context, to lead us through this, we have an exceptional panel where first Tim is going to talk about what's possible. George is going to talk about what are the key barriers. Ken will provide the population perspective. My Bay Area friend Matt is going to provide the entrepreneur perspective. And then Kim is going to provide what's arguably the most important aspect here, the real discussion of the patient impact. So with no further ado, Tim.
Hi, folks. It's a real pleasure to be asked to participate in this August panel. I get to open us up. By way of introduction, I'm a neurologist, a sometimes bioinformatician, and a geneticist at Boston Children's Hospital. I run a research lab there, and it's my privilege to share with you a case that I'm really excited about. It's been in the news a little bit recently, so if you've read about it, please bear with me, but hopefully we'll get to discuss some of the details. But I think the reason I'm excited about sharing this case is because it speaks to a time in the field where our disparate practices, genetic discovery, clinical care, advanced informatics, are coming together in a potentially very exciting way that is really allowing us to actually say this word precision medicine may actually mean something. So without any further ado, let me introduce the case and then move along. So this story begins in January 2017 on a Friday evening. I'd finished a day of work and my wife turned to me at the dinner table and said, would you take a look at this post? This was a post that was put up on Facebook by actually a Boston Children's emergency room physician Jessica Flynn Deed, and she had actually passed along a message from her college classmate who described a family's really terrible plight in Colorado. And this is the story of a family with two beautiful children. The eldest, 6 years old, Mila, had just been diagnosed with Batten disease, a neurodegenerative disorder, without a cure. And they had found one of the mutations in the causative gene, but they needed to identify the second defect for the definitive diagnosis and also to test their 4-year-old son, Aslan. And they were looking for a lab that could help them do whole genome sequencing. And this is something that we had been doing on a research basis for quite some time. And so I looked at the post, and I clicked on the link, and I looked at the website that the family, following likely Matt Mite's instructions, had set up to share their story, and learned about her mysterious decline. This was a young girl who was very, very healthy and energetic and active as a young girl, who then began developing slowly, insidiously, vision problems, gait problems, and then at age 6, accelerating symptoms with worsening gait, falling, speech disruption, slurring of her words, and then using fewer and fewer words, and then stopping using her words. Behavioral regression, and then actually, within the span of a few months, she actually went blind. I learned from that family website that her clinicians in Colorado had given her a partial diagnosis. They had done clinical genetic testing, and they had found a mutation, one mutation in this gene called CLN7, which was a known disease-causing mutation for a disease called Batten disease. What is Batten disease? Batten disease is a rare subtype of a rare family of disorders. They are recessive genetic disorders that are inborn, and they are caused by mutations in one of 14 different genes that control the ability of our neurons to control waste products, to recycle wastes. And when that system goes awry, unfortunately, patients with Batten disease develop vision loss, seizures, motor and cognitive decline, beginning around age 3.5, and then, unfortunately, predictably, with death by age 11. And there were no available treatments because there were really only about 70 known cases of this disease ever published in the literature. So our lab saw this post and said, we want to try to help. And so that led us down a remarkable journey that I'll present to you in very abbreviated fashion. In essence, in the next few months, we performed whole genome sequencing out of our lab at Boston Children's, and we found that the normal CLN7 gene locus here, it did carry the one mutation their doctors had already found, but I mentioned that this is a recessive disease, and I failed to explain that recessive diseases require 2 mutations to manifest. So the question was, where was the missing second mutation? And it turned out that whole genome sequencing revealed that there had been a second hidden mutation found in her non-coding DNA. And this non-coding mutation here, which is inaccessible to clinical sequencing as performed today, turned out to actually be the key second hit that caused her disease. It disrupted the gene splicing, or the assembly of the instruction set required to make this entire 13-block gene, essentially truncating it after block 6 and leaving her with no residual CLN7 protein. So now, as a geneticist and a genomicist, a lot of the times we would stop here. We'd found a precision diagnosis, and we would report this back to the family and say, we've solved your question. But the unusual opportunity that we realized was afforded to us was to ask ourselves, can we actually fix this? And the concept that we pursued was that her genetic instruction set was entirely intact, except for the presence of this mutation here, which caused the cell to ignore the entire downstream half of the gene. And we began reading intently about a technology called antisense oligonucleotides, which had just shown wonderful effects for another neurodegenerative condition called spinal muscular atrophy. And antisense oligos are short 20-base synthetic RNAs that can home in on parts of the genome, and they can essentially do various things, including knocking genes down, but they can also change splicing. And the very simple concept that dawned on us we ought to try to do was to develop antisense oligos that would actually bind to this abnormal site here that was introduced by her mutation, block it, and thereby allow the cell to now continue to read through the rest of the gene. Compressing a lot of work into one slide, we found that we were able to do this. We met our patient in January 2017. We identified the mutation about a month and a half later. We obtained patient cell lines a month after that. And by a month beyond that, we had proven that there was this splice gene assembly defect caused by her mutation. And we began thinking hard about the antisense oligo approach. By September, we had identified and we designed and identified ASOs that actually were working to block abnormal splicing in her cells in a dish. And these were working with a potency similar to that described for other antisense oligo, for the other antisense oligos that had been FDA approved. And then we were able to validate that this actually not only fixed the RNA level defect, but also made her cells healthier. With a lot of assistance from a lot of folks in the pharmaceutical industry who now emerged from the woodworks to help us with this case for this child, we developed a plan to do rat toxicology, manufacturing, and FDA filing in a matter of 4 months. And in January, at the very end of January 2018, we began dosing our patient. A couple of comments. We named it, of course, Millicent in honor of our patient. And this is one of the very first examples of a truly personalized medicine. It is, as far as we know, the first drug created and approved for a single patient purely on the basis of her genome sequence. I think that with assistance, we put together the fastest manufacturing campaign ever for a clinical drug. And we used a rolling FDA submission process whereby we manufactured the drug, began testing it in animals, and before we were done with the testing in animals, we had started injecting our patient because there was so little time to lose. And this whole process has seemed to have paid off. We are now about 1.5 years into the trial, and we've seen signs of disease stabilization and seizure improvement through the course of this 1.5 years. And this, I'm just showing you one of these primary data plots, showing that at the onset of the trial, our poor patient was having up to 30 seizures per day, between 20 and 30 per day. And they were typically lasting 2 minutes at a time, 30 of those per day. Each of these red stars represents escalating doses of drug that we gave to her over the initial dose escalation loading period of our trial. And as this went up, we saw the frequency trend down. And in addition to the frequency, we saw the intensity lighten as well. And by June and July of the first half year, she was having between 0 and 10 seizures per day. And the seizures were not lasting 2 minutes, they were lasting 3 to 10 seconds each. This effect has been stable now over the last year of treatment. We're still adjusting the dosing a little bit to get the frequency a little bit lower, but it's been early signs, but very, very positive signs that this drug is working. So that's the provocative case, right? This is an individualized drug made in 1 year from a molecular diagnosis and then all the way through to a hyper-individualized medicine that we were given permission to proceed with by the FDA. It defined along the way a scientific, clinical, and regulatory path for treating N-of-1 cases. This hadn't truly existed before. We have paths for repurposing drugs, but we don't have paths for approving agents on this time frame when they don't yet exist. And it allows us to even begin to speculate about visionary ideas, clinical genetics, interventional genomics of the future, whereby this type of procedure, this type of drug could be applied and developed to other children with orphan diseases, especially those that fall under the umbrella, under the threshold of what is considered commercially viable. This is something that we've managed to do as an academic lab, and is there a potential to continue doing this in a way that allows us to sidestep some of the challenges in delivering this via traditional industry? And it does raise this theme which was mentioned earlier today. Could drug development actually have gotten to the point where some pieces of it, some pieces of the tool set could be robust enough that we can actually engage them almost under the practice of medicine not under traditional models. So I'm going to stop there. I think I'll leave lots of ethical and legal and social questions for discussion among the audience and our panelists, but set the table up.
Thanks, Tim. That's absolutely extraordinary stuff. And speaking of absolutely extraordinary stuff, our next speaker is George Church, who will talk about some of the key considerations and barriers in this space.
Right. I don't have any slides, so I'll just sit for now. And I would say when we're facing in precision medicine the most expensive drugs in history. We just heard of a $2.1 million per dose drug, and I am assured and I believe that these are cost effective relative to the previous way that these diseases were treated. So gene therapy is cost effective relative to other orphan drugs and all of the other caregiving costs that are, and this is being economically studied. That doesn't mean, just because it is cost effective relative to standard of care at the present, doesn't mean that we're done. My group has spent most of my life trying to bring down the price of many parts of biotechnology. For example, DNA sequencing is now 10 million times cheaper than it used to be, and it was also about 10 million times better quality. So at the beginning of the Genome Project, we were aiming for 1% error rate. Now we're closer to 10 to the minus 9. So this can happen. Well, what are the ways it can happen for these hyper-individualized treatments? Well, hyper-individualized, if you go that far, then you maybe have something that is a practice of medicine and not only doesn't require a full FDA trial, cannot be done by an FDA trial because there's only an N of 1. So that's one way to bring down the costs. Second way to bring down the costs is by eliminating therapy completely. So you can, for some of these diseases, they can be anticipated. You can do genetic counseling. So this is the second part of the revolution here. That we have all kinds of ways of writing our genome or our omes in a therapeutic sense. We can also read them and avoid them at earlier and earlier stages. To some extent, an example of another example of hyper-individualized treatments are cancer, neoantigens. You could be the only person on the planet with that particular antigen that your CAR T cells or other immunotherapies can attack, and that also could be practice of medicine. This is an N of 1. But to some extent, sometimes it's too late. By the time you can detect cancer DNA in your bloodstream, or by the time you can sequence enough to get a neoantigen, or by the time you have a child whose brain has been compromised, it's kind of too late to have a therapy, and your odds are slightly against you. So if you move earlier up, then you might be doing newborn screening or fetal diagnosis or pre-implantation, but even earlier, it can be before you get married. But that's crushing news to find out that the love of your life is not compatible with you. 5% chance, it's about a 5% rate of rare diseases, rare severe Mendelian diseases. That means that 5% of fiancés will get very bad news. You can move even further up to create a list of people with whom you're compatible. Again, we can have an ethical discussion later on. But the point is, earlier is better for preventative medicine, and these can be hyper-individualized and very inexpensive in the sense that the cost of sequencing is close to zero now, close enough that the society can pay for it. It's hyper-individualized in the sense that, and not even FDA approved, in that the cost of false positives for preconception, premarital, predating is close to zero. If I make a mistake where two people are incompatible, just add it to the other 3 billion people that I'm not going to date. So, I mean, it sounds funny, but this is oddly powerful medicine that is oddly not really part of the FDA. So I should answer the question of the whole meeting, which is, can AI accelerate precision medicine? And to show that I have a counterpoint to my own point, which is, yes, we're using it routinely to develop better gene therapy vectors. Like we've made 1.2 million AAV, adeno-associated viruses, which is one of the main gene therapy delivery. We made them custom by using AI machine learning, and it's working spectacularly. I'm actually embarrassed how many years I spent on energy minimization methods, which are now completely swept away by these methods. But anyway, so I can at the same time advocate gene therapy and avoiding gene therapy.
Look forward to your talk next year on G-date.
Thanks so much, David. Let's see. Awesome. So I feel very strange standing up here. My name is Matt. My background is in behavioral economics. Took AP Bio, so got about as far. I connected with Matt's story this morning. So I was out in California because I took a job with Peter Thiel, working at his hedge fund, and was going along doing that for a couple of years after graduating, basically trading currencies until my dad was diagnosed with glioblastoma. So what brought me to this podium today, and I really will try to also not have just the tech founder in the hoodie, which I'm not wearing, sorry, perspective, but also the patient's perspective, because Notable Labs, the company that I started, was really a last-ditch effort to save my dad. And so the very brief version of that founding story is that when he was diagnosed, he had 3 separate brain tumors. And for those of you who have dealt with trying to enroll a loved one or yourself into a clinical trial, they have very strict inclusion and exclusion criteria. And unfortunately, multifocal disease is an exclusionary criteria for most of the drugs that were being developed at the time when my dad was diagnosed. So immunotherapy was out. Any kind of targeted therapies for GBM were out. What was left were all these drugs that were being repurposed. And we've heard a little bit about that today. An example of this was a drug for pinworm that was being developed at Hopkins for brain cancer patients that had responses. There are lots of other drugs around the world, so about 50 of those is the list that I ended up with. And I went to his clinician in Rochester and I said, okay, there's 50 drugs being repurposed for glioblastoma as single agents, and my parents don't want to travel to get those drugs, so pick 3, right? Like, let's combine them together. I was of course met with like who the hell are you? This is not how we practice medicine, which made total sense. And so I went looking for a way to rationally combine those 50 drugs. And so what I ended up backing into is this field that's been around for a very long time. Traditionally has been called chemosensitivity. So we put some drugs on some cancer cells and then we try to predict who's going to respond and who won't. Has not worked very well and in the past. And when I went out there looking at the companies that I could use for my dad's case, all of them had one-size-fits-all solutions. So it didn't matter whether it was breast cancer or brain cancer or pancreatic or whatever. Yet the advancements specifically in brain cancer biology and translational models had gone far beyond what was available commercially. So here at Dana-Farber or at UCSF, there were some really interesting models being employed, but they were not available commercially. So that was really the impetus behind starting the company, was to hire a postdoc, start a lab across the street, have a surgery for my dad, get the cells out, do the drug testing, treat the patient. Worked in terms of we created the models, we found drug combinations. Unfortunately, the turnaround time that it took us to do that was time that my dad didn't have, and so we lost him very early on, and all of the other brain cancer patients that we tried this in. So we needed to move to a cancer type that was going to be addressable much more quickly, and so we are now focused entirely as a company in blood cancers. So our general process is that we're taking peripheral blood or bone marrow from these patients. We are screening thousands of different drug combinations in a fully automated laboratory. I appreciate all the conversations today about scalability. we rank order the drugs that are active based upon specificity. And so what I mean there is that do the drugs target the patient's leukemia cells relative to their healthy white blood cells? And then we do all of that in a time that is actionable. So we're targeting always doing things within a week. Since this was a panel on hyper-individualized treatment, I wanted to talk about a case, which is a public case. This was presented last year at a pediatric cancer conference called ASFO. A patient, a 16-year-old with acute myeloid leukemia that had a bone marrow transplant from his sister, and unfortunately relapsed 6 months after that transplant, which is a very poor prognosis. He was being put on a standard treatment as maintenance, and while he was on that therapy, the clinician sent us his sample for drug testing, and we found that combination of 2 drugs which are approved for multiple myeloma, a different type of blood cancer, were active in the sample. And so when the patient progressed on that standard treatment, they decided to implement the multiple myeloma drugs. And the only reason they were able to do this is that, fortunately, that combination had been tested in kids before, right? And so this is a big barrier towards hyper-individualized treatments is, do we expect that we need to have seen that in a clinical trial before we treat patients? In this case, that was able to happen. And 2 weeks after starting that therapy, achieved a complete remission that was MRD negative and was very durable, which is why the case ended up getting presented. Moving from N of 1 to N of more than 1 is really important with all of this for us in oncology, and so we've been now doing trials that test whole cohorts of patients. So I'm just actually coming back from Amsterdam where this was presented at the European Hematology Annual Congress. We've just completed a trial with Stanford showing 83 and 85% positive and negative predictive value in an adult blood cancer called myelodysplastic syndrome, and trying to now scale this up to see, okay, does it not only predict responses, but if we use assay-guided therapy, will patients live longer than with standard of care? So I'll stop there and look forward to the discussion on the panel.
And now to talk about really the interface between there's all of this discussion and the promise, the articles, the coverage of Tim and George and everyone that we hear about, and then someone comes in with, oh, I have this diagnosis or I have this concern. How do you sort of reconcile the sort of the promise and then the individual fears? And to discuss some of that, Kim.
Yes, thank you, David. Hi, everyone. I'm Kim LeBlanc. I'm the Associate Director of Research Operations at the Undiagnosed Diseases Network Coordinating Center, which is here at Harvard Medical School. And I'm excited to be able to share a bit about our experience working with patients with very rare conditions in the Undiagnosed Diseases Network, and as David said, what the impact on them will be for these hyper-individualized treatments. For those of you who may not be as familiar with the Undiagnosed Diseases Network, or UDN for short, it is a multi-site research project that's funded by the National Institutes of Health. We have multiple different clinical sites, a sequencing core, and basic research groups that are spread across the country, all focused on diagnosing patients who have been to numerous medical centers without finding answers. In the UDN, the diagnoses that we're making are often very rare. Sometimes there are more common conditions, but we have also diagnosed and discovered conditions that are new to medicine. For the conditions that are established, there may be known standards of care, recommendation guidelines for treatment, and in those cases, the clinical sites will recommend those treatments, but that unfortunately isn't the case for many of the patients who come through the UDN. So for those cases, what are we doing? What can we do? One of the first steps for these cases is connecting them with support groups that may be out there. They may be established support groups. They may just be Facebook pages for a gene of interest. Oftentimes these groups, these families are experts in these rare conditions and can work with clinicians and researchers to collect clinical information that will be necessary for research or therapeutic opportunities down the road. In the UDN, we also, if you're not aware, use social media to try to find other patients with some of these very rare conditions, some of the conditions that are new to medicine, to try to put them on that first step of finding others. For these rare conditions, it also takes bridging the clinical and research divide in many cases. Tim mentioned how understanding the mechanism of Mila's condition was helpful in discovering a potential therapeutic. And for many of the patients that we see in the UDN, it's the same case. We do have a model organism screening center in the network that's able to investigate the underlying mechanism of disease in some cases. We also try to partner with researchers outside of the network who may be experts in the condition or disease pathway in order to learn more about the condition and what treatments may be effective. Matt also mentioned the work that he's doing at University of Alabama, and he is one of the principal investigators of the coordinating center. So something we're trying to do now is scale up the approach that he's using at UAB to benefit more patients in the UDN. And my last slide, importantly, patients and their families are crucial partners in this work. This is a picture of our participant engagement and empowerment resource, our patient advisory group in the UDN. In my opinion, we have to partner with patients and families at every step in the process, from the very early research stages to when a therapeutic becomes a possibility. These people are the experts in their conditions and can collect the important clinical information that's necessary to move all of this forward. So thank you, and I'm looking forward to an interesting discussion.
So I'm gonna start off with a couple of questions, and then I'm gonna turn it over to the audience, because there have been so many good questions throughout the day at the other panels as well. And I guess maybe the first, sort of for Tim and George, is how should we think about reductionism in biology? The idea, I feel like we've been moving from a sense where, oh, we're gonna get a gene, and a gene is gonna unlock the secret to everything. And now we've been going to a more sort of, what seems like a nuanced and sophisticated view that it's not just the gene. But now it almost seems almost like a Back to the Future, where now here's this example where here's a mutation, you really fixed or made a lot of progress into curing someone with a very specific gene. We're seeing other examples with some Mendelian disorders, with hemophilia, with others. How generalizable do you think this type of thinking is for patients with disease?
Well, I think it's healthy for the field to think about things as being complex, but it's unhealthy to ignore the simple solutions as they present themselves. So it might be, instead of one gene, it could be two, it could be three. Yes, there may be environmental factors, there may be thousands of genes involved, but very often the lesson of pharmacology and even genetics is sometimes a small number of gene products will be enough. So, for example, height has huge environmental and genetic components, but nevertheless there are 7 diseases that are treated in the clinic with one gene product. We also are entering a phase where the multigenics, even if it doesn't have a 1 or 2 or 3 gene solution, isn't necessarily that hard, because we can control the environment, and we can now edit up to 26,000 sites. That's our latest record for editing of a human genome. That doesn't mean that we're in complete control of those 26,000 sites or any of the off targets, but it is definitely, it makes you rethink what's feasible in terms of multigenics. At least testing hypotheses is suddenly in a new realm. But I'm very, I think that's what I would say about where we should be how we should be thinking about this multigenics and multi-environmental components.
So Tim, how generalizable do you think this is? I mean, was it one of those cases where just everything went right? Because there's so many implicit factors that were right. Or could this be, with a little bit of work, scaled so that many more therapies are delivered? Where, how do you manage expectations? What are appropriate expectations for physicians, patients, and researchers in the context of the work you presented?
So I think it can't be emphasized enough how complicated this project was and how complicated these efforts are. Clinically, scientifically, manufacturing, from a regulatory standpoint, there are many points of failure. And we were extremely lucky to have things line up in a way that allowed this first test case, this first proof of concept to go through. But I do think that, and getting back a little bit to the question of simple and complex genetics, and is everything going to be, at the root of it, this was a simple single gene disorder. And that's what made our managing of the other human and logistical and systems factors it made it feasible. If it were something more complicated, digenic, trigenic, more complex inheritance, obviously we wouldn't be doing this. But I think that examples like this do provide us an opportunity to develop tools and platforms and gain experience with the interventions that we use. Antisense oligonucleotides are, at the moment, now undergoing really an explosion of interest. I mean, this is a field that has been in existence for 30 years, but only now, in the last 2 years, have the fruits really begun to ripen.
And it was supported with research funding? Because I'm leading to the next question.
Yeah.
I can imagine getting a letter from UHD or somebody saying that, thank you very much for your thoughtful proposal. Unfortunately, this is an investigational treatment which we are unable to support. How do you view this as a payer, Ken, or at least representing a population health perspective? When do you see this as ready for prime time and reimbursement? Or like, how do you think, not the answer to this case, obviously, and you're clearly pleased for the patient, but how do you what's your framework that you think about this type of innovation?
Well, it seems like your question is not one of, what does the funding model look like during a research phase, but rather when you have something that works and is known that it's time for scale, how do you actually move into that phase?
How do you make that decision when it's when the science is a work in progress?
Yeah, that's a really good question. So there's a number of different things around it. The insurance world is actually, there's some complexities in there that are not well understood, but there's decisions that people make up front to say, what is it that's actually going to be covered in the insurance world? If I was to make it about car insurance for a second so that everybody can think about it rationally, you guys have no idea how dangerous it is for me to be sitting up here.
We appreciate it.
Yeah, Zach is very persuasive. When you go and buy your insurance on your car, and it's a lovely 1992 burnt orange Toyota Corolla, that's awesome, and it works for you around Cambridge, and you get into an accident, and you call up and you say, hey, I got into an accident, I'd like to get my car repaired. They value the car, they write you the check, and you move on. It doesn't even occur to us that, well, actually, I don't want to drive a 1992 Corolla anymore. I'd like to drive a brand new 2019 Lexus instead. And so I'd like to propose that since I was a very unique case, that I should actually get this. Well, you could actually write an insurance contract that would allow you to do exactly that. That's not a hard financial thing to figure out how to do it. The question would be is what would happen to all the 1992 Corollas on the road, right? I would submit that probably most of them would run into stop signs or something along the line. In health insurance, you've got some similar decisions that actually have to be made. So decisions around what to cover, those don't get made on a daily basis. Those get made, they bulk up and then they are processed in batch to determine should we actually be covering whatever the condition is or whatever the treatment is that actually comes with it. But to this particular case, when you're thinking about this thing, or a case like this, when you're thinking, okay, so what is enough? If there's evidence that you have something that actually works, then there's a conversation that can actually take place about should this be in the commercial contract or not. Until that evidence is there, putting it into the commercial contract would be inappropriate for those that actually purchase that commercial contract. But once you know that something works, now that discussion can actually start in earnest. And we can say, okay, this is an interesting therapy. And then all the other machinations around it will start. What's an appropriate price, those types of things.
When you hear about, sort of authentically, when you hear about something like this, is your sort of, as a person, you're incredibly excited for the patient, but is your sort of almost, I guess, professional view, wow, this can take this will be incredible thing for the health of so many people potentially and reduce all the costs associated with the burden of care? Or is it more of a concern that it'll kind of sort of maybe work and you're going to be winding up just with huge increased cost? How do you sort of see the burden of this from the perspective of a payer?
Sure. Yeah, the personal one is obvious. It's awesome. I'm a dad. I'm a grandfather. I think it's really cool. From a professional perspective, what goes through my head is to build on something George was talking about earlier. How far back do you go? Professionally, I think about what we do is fundamentally it should be in the disease interception business. And the earlier in that process that we can actually implement good diagnostics and great therapies, the better off society will be. Now, we have some decisions that we have to make around that. And those decisions are really hard decisions. Decisions like, how much are we willing to pay for something like that? So take the new SMA therapy, at $2 million. It is a miracle drug. And to say we can take somebody who would normally die at 1 and maybe they can have a productive life, 50 or 60 years for $2 million, that seems like a steal of a deal to me. And I would sign up for that for my own children or for somebody else's. Maybe I would feel differently if it was, my 80-year-old father who's at the other end, and it's an extension of life by 6 weeks or something along those lines. But there's questions around how to do that. That part's not about is it moral or right or those types of things. The question comes down to how. When you have a risk contract that's built around a 12-month cycle, that's really hard to convince somebody that economically that they can make an investment of $2 million and never be able to actually recoup that. That has nothing to do with for-profit or not-for-profit. That has to do with, from a capital deployment perspective, if it's not there, you run out of money eventually. So we can treat the first 5 people, but nobody else. But then there's other questions around, how should a therapy like that actually get priced for the market? And what's the appropriate thing to go through? I think Tim's done a fantastic job in this case of demonstrating new ways to do things. Those are really interesting questions. But at the end of it all, you have to sort of boil those things down and say, okay, we see something, it works. We'd like work to work for all. And then to put the math into real perspective for people, if we made it financial, if we started to look at a therapy like a $2 million therapy for, probably 350, 360 kids a year. So somewhere in there. So we're in the $700 to $800 million to treat everybody that's born this year with that condition. That, in the scheme of the OLRL system, doesn't seem that extreme. If you divide that across every man, woman, and child in America, it's about $2 for each one of us. All of us, how many people here would willingly take $2 out of your wallet right now to save everybody that has SMA, born this year? Exactly. How many of you would take $2 out of your wallet right now to save everybody that has a rare condition in every single thing? How much would that be?
A lot of money.
7,000-ish rare conditions, and so we're at about $14,000. We think healthcare is expensive today, and that's a societal question. Should we actually go that far? I have a personal answer. My professional answer would be it's a societal question, and let's vote as to, I don't mean politically vote, I mean vote from a bunch of consumers in a room together. How do we want to do that?
Thanks. I really appreciate you taking the time to answer these difficult, thoughtful questions in such a considered way, Ken. I know it was a little bit on the spot, and I appreciate it.
That's all right.
So, Matt, I have a question for you. You sort of started your business, it's sort of an entrepreneur business question, really. You started your business as kind of a hyper-personalized diagnostics service model almost, right? But as it's evolved, it's becoming more of a, as I understand, evolving towards kind of more identifying novel therapeutics. Is that right from what you described? What were the considerations, and were there thoughts about not pursuing sort of hyper-individualized therapeutics as a model, or were there other factors that led to the way your very successful and rapidly growing business is evolving?
No, absolutely. So I would characterize that decision as an and, not as an or. So we continue to believe in the hyper-individualized N of one. So can we identify a drug or a combination for that patient, whether it's experimental drugs or off the shelf? I think where the driver for going to larger numbers of patients is because it's much easier to tune the parameters of your assay. So making a prediction on whether or not the patient's going to respond to that therapy or not is very, very hard to do on an N of one basis, right? You don't have a control. So how do you really know if the optimization that you're doing in the lab is making your product better or worse? And when we look at the history in our space, again, he says all these one-size-fits-all solutions. It's also not just across different. I described different types of cancer as being one-size-fits-all. But even within a cancer, like one specific type of leukemia, we work in AML mostly. 8 drugs approved in the last year. Some work via differentiation, some work, are antibody-based therapies, immunotherapies, all kinds of different things, right? Having one test that measures the activity of all those drugs is extremely difficult because their mechanisms of action are all quite unique. And so by tuning on a drug-by-drug basis, we believe the accuracy is going to be much, much stronger than if we try to take that one-size-fits-all approach. And so that ends up meaning that if we predict the outcome of a clinical trial for a pharma partner or for an academic who's running a study, well, then maybe we can solve this problem of not having enough patients for clinical trials, especially with all of the different combinations that are being developed today in oncology. Because, like, for example, those 8 drugs being approved, what is standard of care in AML is actually now an open question.
And I have a question for Kim, and while I'm asking you the question, maybe people could line up if they have questions for some of the other panelists. So my question for you is, as you're talking to patients who are reading about all of these advances, what has been your experience with the expectations that I feel like there's been a series of issues like this where first the Genome Project and then, STI 570, this precision oncology and an IO and all of these different things where you hear about this great promise that on average isn't going to work for most individuals. What has it been like at the point of impact?
A lot of it is talking with patients and families and seeing where they're at and what their expectations are, and not necessarily tempering their expectations, but presenting what the opportunities out there are. There's so much more available now than there was even 5 years ago, and I think Matt's work is a great example of this, where his team will take any patient and discuss what the correct plan may be. And sometimes that too is describing more of what is underlying that diagnosis, what the mechanism is, and why certain approaches may or may not work, putting that in terms that are understandable for everyone.
Do you feel like people are showing up with any type of grounded expectation, or do you think they're sort of thinking like, TV, sort of like TV level miracles?
We work
or level miracles.
Yes, we work with a unique group of patients. I think in your standard clinical genetics clinic, it may be a lot different, but in the Undiagnosed Diseases Network, people have been through such a diagnostic odyssey already that many don't necessarily come in even expecting to get a diagnosis, let alone a treatment. So I think we're working with a unique group of patients there.
That's a great perspective.
Yes.
Zach.
So I know it's very tough to give patients bad news that things don't work. And I'm question here for Matthew first, which is, how do you feel about someone coming to you with a tumor and you say nothing's really going to work? Because that's going to be true a lot of the times. And I have to say, insurance companies have paid for therapies that we all knew were not going to work, like bone marrow transplantation for breast cancer, which ended up costing a lot of money, torturing a lot of women, and not really changing outcomes. How do you think about that, about being aggressive, doing your best, while not hawking false hope?
That's actually our current obsession inside the company. So I'm really glad that you asked about it. So this is, the term here is negative predictive value, right? If it doesn't work in the lab, it doesn't work in the patient. And so the concept is, you test all these drugs and maybe nothing looks good. That does happen, has happened. and that is, that is challenging news for that oncologist and that patient. Of course, we're not directly treating the patient, so it's not. Those conversations happen outside our walls. But I think that it's from a patient's perspective. If this was again my dad or my daughter or my son, and there was nothing that was available through the drug screen. Well, then at least I wouldn't be using a drug that every drug has toxicity, every drug has cost for that patient, and we could be going to look through other methods in terms of clinical trials or using genomics or things like that. But I think fundamentally, actually, the space that we're working in is going to be better at negative predictive value than positive predictive value, right? If it doesn't work in the dish, doesn't work in the patient, is much easier than works in the dish, works in the patient, right? So, perfect example of this is when my dad was diagnosed with glioblastoma, he was given standard of care because he had, which there is a biomarker for that predicts non-response quite well. There's only about a 10% benefit rate to this biomarker called MGMT with the standard chemotherapy and radiation. Why aren't we doing upfront clinical trials in those patients to give them other agents, right? And so that's an example of a negative predictive value biomarker that the paper came out, approximately 10 years before my dad's diagnosis, yet he was still offered that same standard of care rather than onto something else. So I think actually
sort of a way you're saying
that needs to shift, and that we give different drugs. So it's negative predictive value to standard of care, but hopefully positive predictive value to something else.
So I guess your question was, isn't it kind of gruesome to tell people they don't have any hope, and how confident are you in that? But then your perspective is, well, if something really isn't going to work, better to know that up front so that you can advance potentially to something more effective or something at least different, or to provide a sort of better guided advice.
And we have to bring these kinds of approaches to the frontline setting, right? And that's where the patients are going to be much, much more likely to benefit from a precision-based treatment than after they've gone through 3 lines of therapy.
Thanks. Adrian.
I have an easy question. What should the role of the FDA be in hyperpersonalization, and does AI change that or not?
Go ahead, Tim.
I'll take a first stab at that. So I can speak to our interactions with the FDA around the particular platform that we deployed for our patient. And the FDA has a number of roles. They exist to protect the public health. And when you put that in contrast to the title of the panel, hyper-personalized, hyper-individualized treatments, then there's a little bit of a disconnect. But they also provide, so there's less of a public health impact for some of the treatments that we're discussing. Now, whether you consider, however, the treatment is the drug, the molecule that we injected into our patient, or the process, however, if we now are talking about the process of building a drug for a patient with Batten disease, customized to her mutation, now we're talking about a place where the FDA clearly has purview. And so I think they serve a valuable function and valuable oversight over this type of work. I'll just throw out one other scientific piece, which is not lost on us. The FDA also sits in a privileged position. They are a trusted third party that has access to data from many different pharmaceutical efforts by many different self-motivated companies. And they protect that. They don't share that freely with the public, but they sit on that data and can guide other companies along the way without violating intellectual property towards hopefully a better goal, more efficient goal. And I think that's another critical role that they play in individualized medicine.
And just sort of a quick additional perspective, and I want to get to the next question, but I think an interesting perspective is that for years there was this perception that the FDA is sort of really resistant to innovation, or is going to just block and be super conservative. And over the last several commissioners, multiple administrations of both parties, there's been, I feel like there's been this profound switch, and there's a huge interest in pulling innovation, responsibly, as they always emphasize, but but really trying to make sure I'm pulling things forward. And to the extent that when I give talks sort of like in different pharma groups, I actually think a bigger blocker is some of the sort of conservative mindset within some of the pharma regulatory groups internal to pharma, as an example, than an agency that's really trying to look at new things. Go ahead.
Thank you for your wonderful talks. So my question is, besides and comparing
Could you introduce yourself? I'm sorry, to
I'm
continue
Adam
Zach's
Han,
tradition.
studying genomics of aging.
Okay.
Besides rare disease and cancer, how about hyper-individualized longevity project and aging-related disease treatment? Will this open a new channel and opportunity for hyper-individualized precision medicine?
So I guess the question is, how does hyper-individualized medicine relate to longevity, and which youngster's blood do you want injected?
So yeah, I've done a little bit on this. I think one of the possibilities for longevity is that it is a core commonality that we have in common. In fact, all of us who don't die of people in industrialized nations, about 90% of us will die of diseases that don't affect 20-year-olds. So it could be some common set of mechanisms. It's probably not simple, but it could be common. In which case you don't have hyper-individualized. You're at the other end of the spectrum where you have the first true blockbuster drug in history, which all 7.7 billion of us could take and drive down the costs. That said, the way it's configured right now, the way we treat diseases of aging is extremely fragmented and individualized, and that we might have a little of each. And I'm not sure we want to drill down much more than that, other than to provide the counterpoint that it might actually be hypo-individualized.
Thank you, George. Introduce yourself as well.
Hey, my name is Sam Omari, and I'm an investment banker from Connecticut, and we just started working with health AI companies. And one of the things that we're very interested in is the fact that there are a lot of people that will be very old in the future. So that's going to be a big strain on the hospital systems here in America and across the world. So where do you guys see AI solving that issue or problem? Thank you.
I'm sure it'll help.
Any comments? Oh, all right. So any next?
Actually, so I'm going to take that one and go back to the FDA question too, because actually I think there's some. You asked the role of the FDA, and I think there's some things that can actually change that would be really helpful. So, for example, when we do something today, we publish a trial, and you have a treated group and a control group, and it works on the measure enough that we can roll this out to the public, and it's safe enough, and all those types of things. One of the things I think that would actually help, that would be a little bit of a hybrid, is, and I'm not sure who should actually go in and do it, but actually figuring out exactly who it worked for. Should we be sequencing people? Should we be digging down into which proteins were expressed, and what is the biomarker that actually would indicate that this thing actually does work? Because when you look at some therapies, you have, it works for 25% of the people, but it works really well. And then you have other people, it doesn't work at all. And some of them are cheap, some of them are really expensive. The amount of resource it would create to be able to go in and start using those trials to actually dig in and say, this is who it worked for, and then maybe start figuring out why. I think that's a weakness of how the system operates today.
Well, I think there's a huge amount of interest in trying to identify the actual outcomes and trying to figure out the relevant correlates with the outcomes and trying to figure out who are those populations of patients. I actually worry a little bit that people are essentially on the basis of relatively few numbers of outcomes, they're looking at an infinite number of variables, overfitting the data, and then making sort of a little bit of naive predictions. So I think sharing that aspiration, but trying to do it well.
I'm Isabel Chambers. I'm a high school student interning at Dura Bio. And my question was kind of about manufacturing with personalized medicine. And I know, Doctor, you talked about this a little bit. But do you think in the future that there will be maybe small CMOs in hospitals, and how do you think the manufacturing of the drugs and biologics will evolve, and do you think AI will have any impact on that?
That's a great question. Thank you for asking it.
That's an excellent question, and I think that it's really exciting to think about the technologies that could be deployed at this bespoke local microbrewery kind of way. And there are a couple of technologies that have historically navigated that. I would say that, well, I'm sorry, antibody therapy, not at the bespoke level yet. Now, there aren't manufacturers building antibodies within hospitals for patients, but the level of expertise that we've developed with being able to develop a monoclonal antibody against whatever protein target we're interested in, and then deploy that in a safe way, has really, that capability has really expanded in the last 10 years or so. And it took a long time for that to develop. I think that there are other platform programmable like medicines, like antibodies, that can be used in multiple different ways. Antisense oligos are just one of the examples, another example of that. And with time, the more we invest in those types of reusable drug platforms, the more we'll be able to do exactly what you said.
And I mean, ultimately becoming not quite a commodity, but something that's more closer to that than being exotic. Next question.
Yes, my name is David Platt. I teach computer science over there. My question is mostly for George and a little bit for Ken, and that is thinking of your ability now to look at the genome and predict the possibility of these unusual diseases, especially when combined with a particular other person that requires two recessives. Seems to me that you're now well within the range of something that would actually work in modern society and pay huge, huge benefits. We already get tested for, I myself got tested for Tay-Sachs before my own wedding. It's not at all an unusual kind of thing, and if they're going to stick a needle in my arm once, they might as well look for the rest of them while they're there. The popularity of 23andMe, for example, another. And I can just see this thing becoming routine. Ken, how much would you pay, would your insurance company pay somebody when they turned 18 to go get yourself sequenced so you know who you were compatible with and who not? Are we not in the hands of the marketeers now? Can you see Match.com offering 2 months free when you sign up if you agree to get your genetic profile, and when you wink at somebody they'd say, sorry, no, that person is not compatible. I can very easily see this slotting into society as just something that is done routinely. Do you see things evolving that way?
You want to go first? Do you want me to? Yeah, sure.
It's a natural outgrowth of things we do
Yes,
already.
Ken, the question is, you guys going to cover counsel now?
I think my part was how much would I pay for that?
Yeah,
right?
okay. How much would it be cost effective?
Am I the one getting married?
You were going to pay for the 18-year-old to get this test so that...
You're the guy that wants the Lexus, aren't you?
Yes.
Okay, if anybody sees a burnt orange Corolla, it's his. You know, actually that one comes down to how much would I pay, or how much would an organization pay as a risk holder to actually say, can you avoid some of these types of things?
Precisely. How much would it be worth to you to be cost effective?
If you had a 1-year risk contract, the answer is you would pay nothing. And now I'll help you out on that one. If I have 2 people that are going to be getting hitched in some way or another, and presumably they're getting a test because they're thinking about producing offspring,
yes.
And presumably they haven't produced that offspring yet,
yes.
And let's just say it takes 2, 3, 4 months for that to come about, and now somebody's pregnant. One thing that humans have in common is 9 months of gestation, so that's kind of cool. I just moved you out to 12 months from the time you took that test. So actually, if I have a 1-year risk contract, I'm paying for something that actually has no benefit to whoever holds that risk contract. Does that make sense? So a way of actually saying is, what would that be worth if somebody had a risk contract that lasted for 10 years? That one's actually relatively easy math. If you had somebody for 10 years, you would ask, what's the prevalence of those types of things in general? You would ask, what's the cost of actually dealing with those types of things, and boil that back down and say, you know what? It probably is actually worth it to pay some amount of money. I don't know what the exact math would be, but you'd be able to actually calculate what that would actually look like.
There was a guy here last year, maybe a year before, who was saying exactly that you need to have futures contracts on your own health to make that kind of thing cost effective.
Yeah, it's a different type of insurance contract than what we carry today in health. It's actually, though, it's a fascinating concept because you think about childhood disorders. And it's not just a feel-good thing. From a society perspective, taking care of children is, that's a foundational bedrock thing from a society perspective. There are probably models that we need to explore that says, okay, we have somebody that has a rare condition, we're going to send them into an undiagnosed disease network. Who's going to hold the risk long enough to make sure that this thing can actually work? And I think there are plenty of models that could actually be applied. People ask questions about AI and blockchain, those types of things. Can you put together a risk contract that allows you to say, this transfers and there's a transfer payment that keeps coming back, and that becomes a funding mechanism for dealing with people? I think those things are actually feasible.
There's a flip side of this too, where there's a very interesting JAMA paper that Imran Haque and colleagues published when he was at Council, where he talked about saying, okay, well, things like screening for CF, carrier screening is covered apparently, but screening for rare diseases, whereas if you integrate the long tail of rare diseases, like it's been discussed, it's actually it's less rare than the chances of having a child with CF. But in general, like that isn't covered. So he was sort of making the argument for that type of consideration. I see we're running out of time. I want to make sure we have time for our next speaker.
Ken,
So
thank
join
you
me
for your candor and your the look
thanking the
behind the
panel.
curtain.
Yeah, incredible panel.